So yesterday we watched this animation about photolysis, the first part of photosynthesis. We already discussed the fact that there are other names. We talked about what the names mean. We already discussed where it happens. What we'll focus on now are the steps. You likely have this written down because most of you did this yesterday. The first thing I want to talk about are the chlorophyll molecules. They are the things that absorb light, and like you saw in the animation, they are green. They look green. And one of the questions that you had to answer, if you did the homework, was things about color and what makes the plant look green. Does anyone know why they look green? Yes. They do reflect light, but it's not the other light that they reflect. But he has the right idea. The green light is what is reflected. That means that plants that look green are not using green light to do photosynthesis. They are using any color but green. And green is the color that reflects off of them and that our eyes pick up, so they look green. If a plant did not look green anymore, like what's happening right now, it's because chlorophyll is not present to reflect green light to our eyes. So I just wanted to point that out about chlorophyll. They're green in the animation on purpose because that's what color they look, and this is why. Green light is reflected. You will see in the questions that we'll work on today, there are a couple that ask about photosynthesis happening in different colors of light, but the thing you should recognize, if the light was green, less photosynthesis would happen. Now these chlorophylls come in clusters. I just mean like a big group. They're called photosystems, and in the notes when you see PS, that's photosystem. So PS2 is photosystem 2, and later I say PS1, that's photosystem 1. Now I mentioned yesterday, but I'll remind you, photosystem 2 happens first, which is stupid. I agree, but that's how they're named. Can't change how they're named. What happens, we saw in the animation, electrons get excited. They start jumping around. They move to the center of the photosystem, and then in the animation it sort of looked like they fell out and something caught them. Those are the mobile carriers. Now in the animation, the mobile carriers all had names, silly long names like plasticine and plastoquinone. I'm not going to ask you for their names. If you could recognize that something is going to carry the electrons along, that's the extent that I need you to know. Now these electrons move from photosystem 2 along to a stopover and then photosystem 1. But that's their end goal. They want to move to photosystem 1. As they do this, the energy that those electrons bring with them is what drives the transport of hydrogen. In other words, they are helping to maintain the gradient. We know there needs to be a concentration gradient for chemiosmosis to happen, and the electron's energy is used to keep that gradient there. On the video you saw, as soon as enough hydrogen moves so that it's equal on both sides, ATP synthesis stops. This electron energy movement keeps it a gradient so that it keeps happening. So we keep having ATP produced. Now the original chlorophyll molecules, those are the ones in photosystem 2. They're unhappy and unstable because they lost some electrons. So they get their electrons from water molecules. So I'm going to take a water molecule, I'm going to split it up into electrons, hydrogen ions, and oxygen. And so one of the things that I would ask on a test is, what happens to each piece of the water? Where is it all going? Well, I know where the electrons are going. They're going back to the chlorophyll in photosystem 2 because they lost their electrons. The oxygen diffuses out of the cell. So remember, 2 of them will combine, make O2. The hydrogen They're helping maintain a gradient, but eventually they're going to be added to NADP along with the electrons that had been moving around. So these original electrons that are excited move from photosystem 2 to photosystem 1, and then finally to NADP and help it become NADPH. Let's talk about this. So the second part of photosynthesis is the light-independent reaction. So independent would be the key word. That means it doesn't depend on light. The most common other name is the Kelvin-Benson cycle. Sometimes people refer to it as the dark reaction. And I would prefer that we don't do that because that's implying that it has to happen in the dark, which it doesn't. It just doesn't require light, but it could happen when light is there. So dark reaction is something you might see used, but I would prefer that we use either light independent or Kelvin-Benson cycle because they're better. They're better names. Now, where does this happen? In the stroma. So remind yourselves, a chloroplast is an oval with some circles in it. The circles are the thylakoid membranes. The stroma is the fluid that surrounds them. So not on a thylakoid membrane is what we're talking about. Now sometimes when we're describing the light-independent reaction, we talk about the actual chemical process that's happening. And the term is carbon fixation. Now, we don't mean that we're fixing carbon like we're repairing it. Chemically speaking, when we're fixing something, it means we're adding something to it. I'm going to take carbon dioxide, CO2, and I'm going to fix it to something so that it becomes a carbohydrate, and all carbohydrates have C, H, and O as the basis of their formula. So what's the difference? Obviously, it's the H. I'm going to take carbon dioxide and somehow incorporate some hydrogen into it so that it can become a carbohydrate. That is the main idea of what happens in the Kelvin-Benson cycle. I will tell you some details of how that happens, but this is the main idea. So if I ask you what happens in the Kelvin-Benson cycle, this is what you should say. I take CO2, I fix it, I make a carbohydrate, because that's the purpose of it. So, a couple of points about how this fixation happens. First, NADPH from photolysis. So that's where it's coming from the thylakoid membranes, from the first part of photosynthesis. This NADPH moves into the stroma and releases their H. In other words, it gets oxidized. It had an H, it's going to bring it from photolysis to the light-independent reaction, release the H. It's going to get oxidized. That means if it's getting oxidized, something else is getting reduced. When I take CO2 and add hydrogen to it, I'm essentially reducing it. I'm adding things. Since oxidation and reduction always have to happen together, if something is oxidized, something else has to be reduced, and that's the link. So like I mentioned above already, there are some other substances around, and I'll talk about a few of them in a second, but essentially, I take my hydrogen and my carbon dioxide and I make a carbohydrate. And for me to do this process, it requires energy. Where am I going to get energy from? ATP again, that came from photolysis, from that ATP synthase that we discussed. So NADPH and ATP got made in the first part of photosynthesis. Now I'm going to use them up. So when I look at the overall reaction, they're not there because I made them and then I used them right away. Now the next thing that I'll show you is a diagram of what's happening sort of step by step. And it's like a diagram that's in your textbook, although the one in your textbook is a little more simplified. But you might want to find page 177 of your book, because I'm going to talk about my picture and the one in your book because they're essentially showing the same thing. All right. So my picture is definitely more detailed than the one that's in your textbook. And before you worry, I'm showing this picture not because I want you to memorize every detail, but because I like to illustrate where things are coming from because I think it helps it make more sense. In your textbook, you have a sort of a circular picture divided into 3 parts, and I'm going to talk about my diagram with the same 3 parts. This essentially will be part number 1. So if you're writing things down, I would look at the picture in the textbook as sort of the basis for what you're doing because there's sort of 3 parts. In the first part of the Calvin-Benson cycle, you'll see up here it says I have 3 molecules of CO2. And I'm just writing it a bit bigger so that you can see. Each CO2 has 1 copy of carbon. Over here, I have 3 molecules of something that will abbreviate RuBP. That's what your textbook calls it. Its actual name is ribulose 1,5-bisphosphate, which is kind of a long, ridiculous name. So we will always abbreviate it RuBP. Each RuBP has 5 carbons. So I'm writing down how many carbons there are. I'm not going to ask you on a test exactly how many carbons there are, but it helps illustrate what's happening. And for some people, having numbers makes it a little clearer. If I add this up then, here I have 15 carbons. Here I have 3. The first thing that happens is that these molecules all run into each other and do some rearranging. Over here, I have something called 3-phosphoglycerate. We will abbreviate it PGA, phosphoglycerate. And this molecule is arranged slightly differently. I have 6 copies of it. Each of them has 3 carbons. So all I did in this first step was I mixed a bunch of molecules with carbon together and rearranged them. Instead of having 3 molecules that have 5 and 3 molecules that have 1, for a total of 18, I kind of split them up. And now I have 6 molecules that each have 3. So I have the same number of carbons, I've just rearranged them. So this step, if I had to give it a name, it's the rearranging step. I'm adding CO2, it's coming in, the plant has absorbed it, and now we're just rearranging it into a different type of molecule. The second part, so in your textbook, the second part of the picture shows how we get from that molecule, PGA, to something that could give me glucose. First, you'll see that I use ATP. In other words, I'm going to get some energy. ATP will come in, lose its 3rd phosphate like we saw that it does. There will be a molecule whose name I will never ask you. Then I will use, or like I said before, oxidize NADPH. That's giving me a hydrogen That's giving me some electrons. So I'm going to use ATP for energy. I'm going to use NADPH to get some hydrogen and some electrons. And I'm going to end up with this molecule, G3P. It's actually glyceraldehyde 3-phosphate, but I think you'll agree G3P is much easier. I still have 6 copies, like I had before. So I had 6 copies of PGA. I have 6 copies of G3P. It still has 3 carbons in it, so I haven't done anything with the carbons, but I've added energy and I've added hydrogen to it. Now here's the point where 2 things happen. Of those 6, 5 of them keep going in the cycle and 1 of them leaves. So 1 of them leaves. G3P has 3 carbons. That's not big enough to be glucose. Glucose is C6H12O6. That means that this cycle happens 2 times for every 1 glucose that I make. So every time 1 photosynthesis reaction happens, this process happens twice. It makes sense if you look up here. I only used 3 CO2s, and if I look at the reaction for photosynthesis, I know it starts with 6 CO2s. So I have to do this cycle 2 times. So one of these G3P molecules leaves. Once I have 2 of them, I can make glucose. The other 5 rearrange again, and we would call this process regenerating. I'm regenerating RuBP. That was sort of the starting point of the cycle in the first place. But I take those 5 molecules that each have 3 carbons, and I use some energy, and I rearrange them so that now I have 3 molecules that have 5 carbons. Same number of carbons, they're just rearranged.